Hi students, it's Mrs. Belliston. This is a video for your Science 7 end of year review. It's going to be a huge help for completing the review page because we are going to be going over all of those questions. This end of year review sheet is due on Friday, April 28th, 2017. I've emailed you the assignment a few times, so you can find it in your email. There are a few options for turning it in. You can fill out the assignment in Microsoft Word and then attach it to an email and send it to me when you're done. You can also print it. If you like actually filling it out and writing it in, then you can take pictures on a phone or a camera and attach those pictures to an email and send it to me. You can also just email me the answers 1, 2, 3, 4 in the body of the email and send it my way. Basically, I want this assignment from you. This is your only assignment for the month of April, so it's worth doing. The first question asks you to compare and contrast atoms and molecules. Well, my first question is, do you know which one is smaller and bigger? Well, let's talk about it, and I'll always be drawing a few pictures, at least doing my best to. So, atoms are the smallest unit of anything that's a substance. So you can have atoms of carbon, you can have atoms of, oxygen, right? So let's draw some oxygen atoms as well. Okay. So we have oxygen atoms as well, right? Now, I'll just put down here oxygen for short. Now, these are atoms because they are by themselves, right? So atoms are when it's only one thing, like pieces of Legos. Now molecules are when you start to build those things together, all right? So I could take carbon, okay, and I could attach a carbon atom, okay, and then I could take these two oxygen atoms, okay, and I could put those together, and that would actually make CO2, carbon dioxide, right? So this right here is a molecule, and atoms are when they are by themselves. All right, this one I think you've got. I think this is easy. So it says to draw the different molecule arrangements for a solid and a liquid and a gas. And it wouldn't be appropriate to talk about this without dancing a little bit, right? So remember that the molecules in a solid are still moving, right? The molecules are really close together. So you can draw those molecules. They're in a structured lattice. I mean, so those molecules, and now I'm drawing them very large because of my pen. But remember that these molecules are, so small that not even microscopes can really see them. But they're all very, very close together. And can you show me how those molecules move? They vibrate in space, right? So the solid maintains its shape, like think of a solid phone or something. It's maintaining its shape, but don't forget that those molecules are still moving. A liquid, how about those molecules? Are they farther apart or are they even closer together? They're farther apart, right? So you can draw dots that are farther apart, and these are free flowing, right? Liquid, think of water. All the molecules in a water cup are just free flowing around. Now a gas, we refer to these as the crazy molecules, right? Because they are moving around like crazy. So you can draw molecules with, sorry, this is kind of hard to do with a pen, but with kind of like tails behind them, because they just do whatever they want and they take up all the space that they possibly can. This question asks, which of the states of matter have volume? Which of the states of matter have shape? So you'll have to bear with me and my pens that I am able to use here, but let's first talk about shape. Okay, so can you guys think of the solid, liquid, and gas? Which of those things have shape? In other words, if they're sitting on the table, which have shape? Does a solid have a shape? Does a liquid have a shape? Does a gas have a shape? I hope you guys are pretty clear that the only one that has shape is a solid. Okay, so only a solid has shape. Gas and liquid do not, right? And that is shown by when you like pour, you have a cup of water and it's in the shape of the glass because liquids take the shape of the container they're in. And if I pour that water out, it's not going to stay in that shape of the cup. It's going to then take the shape of the countertop and spill everywhere. How about volume? Volume is how much space something takes up. So if I have my, I'll just show you my globe. Right here, my globe sits here. Its volume, it's a solid. Its volume is set. It's not just going to sit here and magically expand. That would be pretty cool. But solids have a volume. In other words, it's a amount of space that's going to stay the same. Okay, so definitely a solid has volume. Okay, let's think about a liquid and a gas. Do those have volume, like a set volume? Well, a liquid does, right? If I set a cup of water on the counter, that cup of water isn't going to, the water inside isn't going to expand or contract. Now, the water can evaporate, but that's something different. We're just talking about in the moment, does something have a set volume? And so yes, a liquid also has a set volume, okay? And I want you guys to take the time to write it out nicer than what I'm doing, but that'll give you a chance to kind of make sure you really know it. So you can write out solids and liquids have volume and solids have shape. You can write that out. Gases, let's just take a moment to talk about a gas. Okay, gases are the wild child, okay? Notice that gas is not on either of these lists. So gases have no volume and no shape. This is seen when you bake cookies in your house, right? Does that gas of the cookie smell stay in that oven? No. It goes anywhere it wants, right? It goes anywhere that it's allowed. So anyway, just everywhere. So the volume of a gas is not fixed. It just is always spreading because those gas molecules are absolutely crazy. We sort of touched on this in the last question, so there was a hint. Why is it that gases coming from your oven in the kitchen can be smelled in your own room? So why is that? So we have an oven, we have cookies inside, or maybe we'll have a purple cake, and these smells just go everywhere, right? It's because gases have no volume. Gases move anywhere where there's space to be moved. So gases, molecules are the fastest. They move like crazy. They have no volume, no shape. So because of that, gases move everywhere, and you can smell the good things as well as the bad. Great question. What happens to molecules when they heat up? Well, let's pretend I'm a water molecule, shall we? That's fun. I will be a water molecule starting in an ice cube. Me and my molecule friends are just vibrating in space. That's all we're doing because we're in a solid. But let's say someone takes me, water molecule, out of the freezer. Okay? Watch what happens as I get out of the freezer. Okay, now I'm a liquid. I've just melted. I have more energy, okay? I have more energy and I take up even more space probably. So I'm a liquid. I'm a liquid and I'm moving faster. Okay, so molecules, when they heat up, they move faster. Oh my goodness. I've melted, but it's getting hotter. And oh my goodness, it's getting so hot. It's getting so hot that I'm turning into a gas. So what happens to molecules when they heat up? Well, they move faster. All right, had to take a drink break after that one. What happens to roads here in Utah during the winter versus the summer? Why would builders build spaces into roads and buildings in places like Utah? This actually has a little bit to do with the last question. I want you to think about what happens to molecules when they heat up. They speed up. And they also, because they're speeding up and they're hitting off of each other more, they sort of make things expand, okay? I don't know if you've ever in the summertime been outside playing a lot and your fingers get like kind of swollen when they get really, really hot. I know that in the summertime my ring is a lot harder to get off, and right now in the winter and the spring it's a lot easier because it's colder, and so the molecules in my body aren't moving quite as quickly and they're not taking up as much space. So what happens to roads in Utah in the summer? Well, in the summer, the roads slightly expand. And in the winter, what do you think they do? They slightly contract. So why would builders have any interest in this? Why would builders, if you're making a road, they build like tiny, I'll kind of draw it with black. It's kind of tricky, but in between a lot of the lanes on the freeway, you'll actually see tiny, 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 tiny cracks. If you look out the window and look down at the freeway as you're driving, you'll actually see these tiny little cracks that they've built into the freeway. Why would that be? Why would that be smart in a place like Utah where we get quite warm in the summer and quite cold in the winter? Well, it's to allow that material some space for expansion and contraction. Number 7, fill out the following table. We're shifting gears just a little bit. We're going to begin talking about mass and talking about volume. Two very important concepts. But first, let's define them. Let's make sure you know what they are. What is this? So I often like to talk about and show you a balloon and maybe a tennis ball, okay? Because these very clearly help us kind of define the difference between mass and volume, because they are different, okay? Mass is how much matter something has, or we can also define it as maybe its heaviness. Sorry for my lousy handwriting. Heaviness. So, as I sit and hold these two objects, if you can imagine holding them yourself, which one is heavier based on what you know? Well, definitely the tennis ball is heavier. So the tennis ball has more mass, okay, because it's made of, it has more matter contained in it. Now, volume is different than mass. Volume is all about how much space something takes up. Okay, so let's see how, I'll write in here, how much, wow, I really need like a little drawing board thing, how much space something takes up, okay? So based on that, which of these takes up more space? Definitely my cool pink balloon, right? So the balloon has more volume, but the tennis ball has more mass. So let's think about some units that we could use if we were going to measure mass. Now, I don't want just any units. I want metric units, right? These are units that the whole world uses. So how would I measure something like the mass of something in metric units? Well, there's a big one. It's grams, right? And it could be, you could put different things on the front of that. It could be a kilogram or a milligram, but the basic word there, the basic unit is grams. Now, how would I find that? Let's go ahead and go to this bottom question for mass. What instrument would I use to find that mass? Or how could I find the mass? Well, I could use a scale, right? We have digital scales, like where you just set something on it. I mean, you guys have seen those if you've ever weighed yourself. But I would find the mass or the heaviness of something based on, you know, putting it on a scale, and it would tell me how many grams. Volume is a little trickier because there's a few different ways to do things. So let's first talk about possible metric units. Well, it depends if we're talking about a liquid or a solid. So when you guys go to the store and buy soda, oftentimes we buy, how much soda do we buy? Go pick up it's kind of that tall bottle, a 2 liter, right? Liter is the volume unit for liquid. So L-I-T-E-R. You're going to have to write better than me. But that's for liquid, right? So I'll just put L-I-Q to just kind of show that. Now, but also, I mean, this, my balloon I'm trying to pick up here. Wow, it's being really challenging with me. I don't know if I'm going to be able to get it. My balloon also has, it's not a liquid. I can't pour it into a cup and see how many liters it has. So I could do a few things. If I wanted to find, I could also find it. Oh, I'm kind of actually getting into this question here. So what instrument could we use? We could use like a measuring cup, right? If it's a liquid. M-E-A-S-U-R-E-I-N-G, measuring cup. But we could also use like a meter stick, because there's a formula to calculate volume. It's length times width times height, right? So if I find length times width times height, if I'm doing that in like centimeters, I'm doing a centimeter times a centimeter times a centimeter. How many centimeters am I multiplying together? Well, 3 of them, right? So another unit we often see is centimeters to the 3rd power, or meters to the 3rd power. So these are other units that you might see as possible metric units, right? And then, so what instrument could we use? We would be using like a measuring stick or something. So if you could bear with me as I write the rest of this, please write better than me. And you know, so that's filling out this table. This was definitely one of the longer questions, but yeah, maybe go take a break for a minute. So here we have the option to actually look and see the volume of some of the following substances. So remember, volume is how much space something takes up. So let's take a look here at our first example, a liquid volume. So if you were looking at this and you needed to decide how much liquid was in this measuring cup, remember this is probably going to be in, I guess you could, as long as it's in some kind of metric unit, I should have specified, but we'll say it's in milliliters. So how many milliliters do you see in here? Well, you got to kind of look at the tick marks. Pay attention to the tick marks, right? So you see that it's of course up to 40, but there's even more than 40, right? What's this line right here? Well, that is 50, right? So where we are at is maybe about 47. Now that may not be perfect, but it would be pretty close. So 47, and I said to include units in your answer, and I know I didn't actually tell you what the units were on this. So we'll just say 47 milliliters, lowercase m, capital L, right? 47 milliliters, or you might say 46, because you can't totally tell because you're just guessing in there. All right, how about a solid box? Remember, I gave you a formula on the previous slide telling you how you could calculate the volume of something, how much space it takes up. If it's a box, you can do the length times the width times the height. So I believe you will have a calculator on the SAGE test, but a box with the length 4, width of 10, height of 2 centimeters. So the math here is 4 times 10 times 2. Okay, so 4 multiplied by 10 multiplied by 2. So you can just break it down into smaller math problems. So what is 4 times 10? It's 40. And then we do 40 times 2, which is 80, right? Now 80, but I want units on all of your answers. Remember that we have multiplied 3 different centimeter units together. So we have a unit to end with of centimeters cubed. Now, there is one more way that we can find the volume of something, and it is through, you know, this method called water displacement. So let's say we have a really awkward object. I'm sure I have something around here. Let me grab oh, I know. How perfect. What if I wanted to find the volume of my glasses? They're super awkward. I can't pour them into a cup to get, it's not a liquid, and it's not a box. I can't take the length and the width and the height of these. It's awkward, right? So what we can actually do is something called water displacement, where I take a cup, like something like, or a beaker, something like up top, and I fill it up with water, because it's called water displacement. So let's say I take, I fill it up to 50. So the volume of the water before I drop the object in is 50 milliliters, right? 50 milliliters. And then I drop it in, and after I drop it in, the water is going to go up, right? Just like when you get into the bathtub, the water goes up, right? Because your volume displaces the water. So I drop my glasses in, and now the volume reads 57 milliliters. Okay, so it was at 50 and now it's at 57. So what is the volume of my object? Well, it's the difference. If it was at 50 and it went up to 57, what's that difference? I hope you can do 57 minus 50 to get 7 milliliters, right? So the answer here is 7 milliliters. That is the volume of these glasses found through water displacement. I hope this one is easy because of the last question. Describe how you find volume using the water displacement method. But I can explain it again. Get a cup. Fill it up to a certain amount of water that you know. Then drop the object in and see what the new volume is. The difference between the original and the new volume is going to be the volume of that object. So hopefully that makes sense based on what we talked about on the previous slide as well, to find the volume of an awkward object. All right, I got to keep my glasses on for this one. Number 10, define density. What does it mean for something to be dense? So there's a few different parts to this question. So yes, you'll want to write them out. And yes, I want you to actually write them out better. It's really awkward for me to use my my pen. So you will need to write them out better than I do. What does it mean for something to be dense? Okay, and letter B, what is the formula for density? And letter C, what are the units for density? All right, so let's start with letter A. Define density. What does it mean for something to be dense? Well, I have often talked about the difference between a cake, a piece of cake, and a piece of brownie. Which of those two things is more dense? Definitely the brownie. What that means is that the material is more compact. So there's more things packed in. So density can really be sort of, what we mean by density is how packed the molecules are, or the material is. So I cannot write all of that out. We'd be here forever, but you can sort of write out, you know, how packed the materials are. Okay, so you can write that out. How packed the molecules or materials are. The more packed they are, the more dense they are. The less packed, the less dense. Letter B, what is the formula for density? Please, please, please tell me you remember. And please, if you don't, maybe this will help remind you. Oh my heavens, do you remember? So we often say that density equals love. And we do that because it helps us remember the formula. If you draw a heart and you cut that heart in half because sadly a lot of love is broken. What you see on the top of that broken heart is sort of an M. Do you see it? Do you see it? I'll write it in purple. Do you see that M? So M divided by, right? It's kind of like a divided by a fraction sign. And then what letter do you see on bottom? I hope that you see this V. So M divided by V is how we calculate D, or density. Now we've just spent the last couple slides talking about mass and volume. So now we're relating the two things, and how they relate is that mass divided by volume equals density. So that helps us to sort of help on letter C talk about the units. Okay, so all you have to remember, well, what are the units for mass? We talked about grams being the unit for mass, right? So the units for density are going to be mass over the volume units. So grams over, what are some units that we decided would be good for volume? You can go back to your little chart a couple questions ago. Well, we said that milliliters, or so there's a lot of different units. It could be grams per milliliter. It could be grams per liter. It could be kilograms per liter. I mean, it doesn't matter so long as you have a mass unit over a volume unit. And it also could be grams per centimeter cubed, right? Because centimeter cubed is also a volume unit. So let's take some practice actually doing some of these density problems. So calculate the density for an object that has a mass, m. Okay, so remember our formula, density equals love. We want to do mass over the volume. So we want to calculate the density for an object that has a mass of 100 grams. So 100 grams divided by has a volume of 10 centimeters cubed. Okay, so the volume is 10 centimeters cubed. So 100 grams divided by 10 centimeters cubed. Do you know what that equals? 100 divided by 10? Well, how wonderful. It's 10, right? And don't forget to include the units in your answer. So remember, the units are exactly what we see here, okay? It's grams over centimeter cubed. So your units will be exactly that, 10 grams over centimeters cubed, or to the third power. Don't forget, I mean, these units, if you ever get confused about, well, wait, what are the units? Just remember to look that it's mass over volume, grams over the mass unit over the volume unit, grams per centimeter cubed. All right, we're going to bump up the work a little bit on this one. Calculate the density of a box that is 30 grams. So once again, I just want you to remind you of our cute little density heart. The formula for density is mass over volume. Okay, so we have m divided by v. So, but here we go. So the box, we're calculating the density of a box that is 30 grams. So 30 grams, that is the mass unit, 30 grams. Got it? And the box, we don't know the volume quite yet, right? We don't know the volume quite yet, but look, we have some dimensions of a length and a width and a height. So how could we calculate? We need to calculate this V, mass over V, volume, the amount of space that box takes up. So how could I calculate that V? Well, I'm going to do length times width times height, right? So it'll be 30 grams over 3, and I want you to show your work on this one, so please show your work, 3 centimeters times 1 centimeter times 5 centimeters. I'm getting better with my pen, wouldn't you say? So what is 3 times 1 times 5. We'll just break it down into smaller multiplications. So tell me what 3 times 1 is. 3. And then multiply that 3 times 5. 3 times 5 is 15. So let's go ahead and take, now we have 30, we still have the 30 grams, but we're going to divide it by 15 because that was what was 3 times 1 times 5. And remember, it's 15 centimeters cubed. Why is it centimeters to the third power? Well, because we multiplied 3 centimeter units together. Okay, so 30 grams, we're now right over here. So look right here, 30 grams divided by 15 centimeters cubed. Do you know what that is? 30 divided by 15, well, you'll probably have a calculator on the Sage test, so definitely put it in there. 30 divided by 15 is 2. Okay, so that's 2, and do not forget your units. Always include your units, and if you're confused about them, just look at the way they are right here. So it's 2 grams per centimeter cubed. Okay, thank you. Okay, friends, we're back to the awkward object that is dropped into a graduated cylinder. So we are looking for density again. So there's a little bit of math involved in what we do this year. So don't forget our formula for density. Always remember the heart, mass divided by volume. So we want to calculate the density of an awkward object, maybe my glasses, that is dropped into a graduated cylinder. So it's that same kind of water displacement method where it's awkward. So I can't find the length and the width and the height, and it's not a liquid, so I can't pour it to see how many liters it has. But I can find how many liters it will, like how much water it displaces. So here's our formula. So density, we are looking for density. We want to calculate density equals mass divided by volume, right? What is the mass of this object? Did they give it to us? Look at the screen. Did they give it to us? Yes, they did. They did right here. It's 14 grams, right? So the mass is 14 grams. So I'll put that here. And what's the volume of that object? Well, we have to kind of figure it out, right? So they gave us a couple of important things. They said that before we dropped the object in, it was at 50 milliliters. But then I dropped the object in and the water went up to 57. So tell me, what is the volume of just the object? Well, it's the amount that it went up, right? Because when I put these in there, that's what went up. So that tells us the volume. So what is 57 minus 50? And we actually did this a couple of slides ago, but it's 7. So that's 7 milliliters, right? So 14 grams divided by 7, milliliters. Do you know what 14 divided by 7 is? If not, you could use a calculator. 14 divided by 7 is 2. So 2, and I always want units. Units are important. So it's 2, and then if you're wondering, well, what units is it? Always just copy exactly what you see here. So the units are 2 grams per milliliter. Cool. All right, this is something we didn't talk about too much earlier in the year. It's just we covered it in our classes and our reviews. So let's definitely cover this one nice and good, just in case you haven't come to the review classes. So the Earth is composed of many substances. The question asks, what are the Earth's basic layers, starting from the outermost layer and moving to the most centered layer. So I'm going to try my best to draw some circles here, and I'm sure it won't be gorgeous, but let's bear with me. So let's start with the outermost layer of the Earth. Do you guys know what it is? And you guys will write it, maybe rather than draw it, or you can draw it. The outermost layer of the Earth is called the crust. Okay, this is where we live. It's where the mountains are. We live on the crust, right? The ocean water sits on top of the crust. Let's go ahead and draw the next layer here. Well, let me just kind of draw. I'm just going to draw this big orange stripy layer here I'm drawing. This is all, do you know what it's called? I know you've learned this in probably different classes, but this is called the mantle. Okay, it's kind of a thick, really really slow moving substance. Do we ever get to see the mantle coming out of the earth? Do you know what I'm getting at? How about those things that go volcanoes, right? So we do have occasions to see the mantle coming out of the earth when we have like a volcano. Yeah, and I think I should draw a volcano. So yeah, here's a volcano for you right there. So we get some of this mantle coming up out of the earth sometimes in the right places. So what is in the very center of the earth? Well, let's draw a couple, let's draw it. So what is this purple middle part? What do we call that? Do you know? Let's make a thicker marker here since I'm using thicker markers. Well, the very center of the earth is called the core, just like you have like the core of an apple. And sometimes people will break up the core into an outer core and an inner core. So if you see that, you may see that sometimes where there's like kind of two separate cores, outer core and inner core. But these are the three basic layers, starting from the outermost is the crust, and then the mantle, and then the core. And we could compare this to like an apple or something. If we were to compare it to an apple, what would the crust be on the apple? It would be like the peel, right? Where it would be, if we compared this to an apple, what would be the mantle? The yummy apple stuff, right? The stuff that you like eat. The white part of the apple could be like the mantle. And then what would be the core of the apple? Well, I think it would be the core. So, yeah, the core of the apple would be like the core of the earth. All right, we are halfway through the questions. So if you haven't yet, I would recommend taking a break. But this question's great. Number 15, why are there layers of the earth? So we just talked about how the core is in the center of the earth, and then the mantle, then the crust. What sits on top of the crust? Well, it's water, and yeah, the crust is like all of our land and mountains. But then what sits on top of the water and the crust? What's all of this around us? It's air, right? So air even sits on top of that. So we have all of these layers in the earth. And why are there layers? Well, the big word, do you know, it's density. The most dense things on our earth are at the core, because the core is the center of gravity, right? All things on the earth like pull towards the center. And so the most dense part of our earth is the core, because more dense things are going to sink and less dense things are going to float. Just like if I had a big tank of water, if I had a big hunk of metal and I put it in the water, that metal will sink, right? And if I had a big chunk of wood, wood is less dense than water, so wood will actually float, right? So why are there layers of the earth? Well, it's because of density. But I want you to write a more detailed answer, okay, in explaining that the core is the most dense, which is why it's at the center of the earth, and slowly things get less dense as you move out, and our air is the least dense, which is why the air is sitting on top, right? So things always layer according to density. If I were to make like kind of like a big cup full of different kinds of liquids, they would layer out according to density. And you might even go check in your refrigerator right now, and you probably have like a salad dressing that has layered a little bit according to its density. So For example, let's say I put some corn syrup in there. Corn syrup is very dense, so it would be at the bottom, right? So things layer according to their density. And then on top of that might be water, right? Maybe I would have water that would layer on top of that. And then on top of the water might be some oil that I poured in, okay? So why do these layers not mix? Well, it's because they have different densities, right? So the oil sits on top because it's the least dense, right? So things layer according to density. You see it in a cup in your kitchen, in the salad dressing in your fridge, and we see it in the layers of the earth. Number 16, define the function of the following organelles in the cell. Well, let's first look at nucleus. Do you guys know what a nucleus is? Well, let's draw a cell down here. Anyone know where the nucleus is? What it looks like? Well, I will draw one for you. It is a region in the center of the cell that contains all of the cell's DNA. It is the control center. Okay, so you can write some different things down here, but let me write here. I'll put, I have to really put short things since my pen is awesome, but I'll put it's the control center. In other words, it has all the DNA. So I just think you should put that, all DNA. And that DNA tells the cell what to do, right? So this here is the nucleus. I'll just put an N for nucleus. Now, what about the cell membrane? What do you guys think about the cell membrane? Where is it? It's already drawn on this cell that I've just drawn below, but it is right here. It's the purple outline, okay? It is this whole outer layer, right? And I want you guys to always think of this just like the walls in your house. It's like a protector for the cell, and it's going to let certain things in and other things it'll say no way, and it's going to let certain things out, like you take the garbage out of your house, you take other things out of your house, and you bring certain things in. So think of the cell membrane just like the walls of your home, okay? So this is the cell membrane. So what is its function? It's the protector, okay? And it's also like the bouncer at a big club. Wow, you cannot read that. Protection, but then also you can put that it is let certain things in and other things not in, right? So it's kind of like it's the protector. All right, so the last two organelles that I wanted you to define are two organelles specific to plant cells, okay? So I'm going to draw a plant cell over here. Please notice what's different about that plant cell. Its shape, right? What shape is that plant cell? It's square, right? Whereas the animal cell is more circular. So that's something to take note of, that plant cells are more square. So a plant cell has a cell wall. That's this, it's an extra coating. So they also have a cell membrane. So I'm going to, inside this plant cell, I'm going to draw a cell. There's a cell membrane, okay? So I'll put CM for cell membrane, but it also has this outer layer that's an extra strong, thick layer. So I'm going to make it a little bigger. Ooh, I'm going to make it a deeper green here. This is called the cell wall. It's an extra protective layer because guess what, guys? Plants don't get to come inside and eat snacks and sit by the fire, right? So they have an extra layer of protection that keeps water, like it keeps the water storage in, and it also is like insulation. So a plant cell has two, like it has the cell membrane and the cell wall. Okay, so when you define it, you can say that the cell wall is an extra layer around the cell that helps protect the cell. So it's an extra protector. All right, how about chloroplasts, guys? Chloroplasts are these small green, there's stacks of chloroplasts in the cell. They're kind kind of stacked up like pancakes. And what does that allow the cell, a plant cell, to do? Well, they're green, the chloroplasts are green. The chloroplasts allow the plant to get energy from the sun. So the chloroplast, we say that it converts sun energy. Wow, you guys are really going to have to listen because my writing is terrible. Converts the sun's energy into, wow, into, like, you know, apples or into leaves. Anyway, so it uses the sun's energy to do that. So anyway, a little bit longer there, but the cell was a big part of what we talked about this year. So I want to make sure you guys know these parts here. Number 17, plant and animal cells have many similarities. We just talked about them on the previous slide. They both have a nucleus where the DNA is stored. They both have mitochondria where energy is made, and a cell membrane that is on the outside wall. It's the protector of the cell. Okay, so those are similarities, but there are 3 organelles that a plant cell has, but an animal cell does not. List these 3 organelles and describe their function. So let's start with the 2 that we talked about on the last slide. Do you remember? The first one we talked about was a cell wall. Okay, and do you remember its function? I won't write it out, but its function is an extra protective wall around the cell. Why does it have it? Because plants live outside in the really extreme heat or the really extreme cold, so it just needs that extra protection for insulation and water retention. So you can write, don't forget to describe, write out the function. So cell wall, the other one we talked about was a chloroplast, C-H-L-O-R-O-P-L-A-S-T, chloroplast. And do you remember what that one does? It captures the sun's energy, right? And it turns it into fruit, or anyway, so that's the organelle that makes plants be able to do photosynthesis. And then lastly, the last one I want to talk about is, it starts with a V. It's called a vacuole. V-A-C-U-O-L-E. And let me draw that for you. So in a plant cell, we have this giant plant cell. Here's the nucleus. And let me draw a, we got to draw some chloroplasts, right? There's these like stacks of pancakes. And let's draw an extra thick cell wall around it in a lighter blue. Nice. Okay, so the last one I want to talk about is called a vacuole. Okay, and what it is it's like this giant water storage. So it takes up a good portion of the plant cell, and all it does is store water and extra nutrients for the plant. So it's called a vacuole. It's like a refrigerator inside of the cell. Why is that important for the plant cell? Well, once again, remember that it lives like in outside. So in the summertime, it has to be able to survive, like kind of collect more water and then make that water last. And so when you see plants that get wilty, well, one, it might be that they're dying, but if it's just like a really, really hot day, sometimes plants get wilty, and it's the reason why is because all their vacuoles are shrunk, have shrunk. And so when you water the plant, all those vacuoles inside the cells expand, and then the plant looks like stiff and firm again. So anyway, these are the 3 main organelles that plant cells have, but animal cells do not. All right, we actually even did this experiment, number 18. A drop of food coloring will spread through an entire cup without you stirring the water at all. What is this process called? Well, we actually kind of already discussed this earlier in the video. We talked about how, you know, first off, liquid molecules are always moving, right? So they're always moving. So if I put a drop of red food coloring down at the bottom of this cup, is that red food coloring just going to stay there? No, it's not. It's going to diffuse. And so this process we're talking about is called diffusion. Okay? D-I-F-F-U-S-I-O-N, diffusion. Now, what diffusion says is that particles or molecules move from an area where they are highly concentrated and they move to an area where they have low concentration. So when I drop that red food coloring in my cup, it's highly concentrated right there in the cup. So now it will diffuse everywhere into the cup. And it will make the cup of water, you know, all turn red. So I don't have to stir it, I don't have to shake it. It's just this process of diffusion that allows it to do that. And if I'm doing it, when we did it in class, we did it in a cup of cold water and we did it in a cup of warm water. Where do you think diffusion happened faster? Think about where the molecules are moving faster. Is it in the cold or the warm water? It's in the warm water, right? So anyway, diffusion will happen quicker in warmer water. So if you want to write a definition for diffusion, it's when molecules move from an area of high concentration to an area of low concentration. All right, so we just talked about diffusion. Now let's talk specifically about osmosis. Now osmosis, the big difference is, okay, I'm going to write it out on the board here, is that osmosis has everything to do with, what am I writing? Anyone know? It's like Wheel of Fortune. Water. Okay, so when you see the word osmosis, I want you to think water. So it's the exact same thing as diffusion, molecules moving from an area of high concentration to an area of low concentration, except that osmosis is water, like the water does the moving. So water moves from an area of high concentration to an area of low concentration. So for your definition, you could put that osmosis is, oh, I wish I had like a little writing board, specific for water. Okay. Now, one example we've talked with quite a bit is if we had something like an orange slice sitting out on the counter, right? So if we let this orange slice sit out, there's quite a bit of water in fruit, right? So if I'm letting this orange slice sit out overnight, there's a lot of water in the orange. And so, and in the air, there's not quite as much water. So in the morning, if you looked at that orange, it would be shriveled up a bit. The water would have left that orange. What process made that water leave? Well, osmosis, right? So the water molecules were highly concentrated in the orange. So osmosis says that water moves from where it's has higher concentration and moves to where it has a lower concentration, which makes all that water leave the orange. Okay. So remember that diffusion is with everything. It could be smells, stinky smells, any molecule that moves from a high to a low concentration. But osmosis is exactly the same, except that it's specific for what? Use my chicken scratch, for water. Okay? So this asks you to draw arrows to show the direction of water movement in the following osmosis examples. So remember, we're always in osmosis, we're focusing on the water movement, okay? So we take some kind of maybe like cell or something in the inside that has 10% salt, and on the outside it's saying that it has also 10% salt. So which way does that water move there? Well, in this scenario, the first one, it's the exact same inside the cell as it is outside the cell. So the movement is in both directions the same amount of movement. Remember, water is always moving. The molecules are always moving. and everything. But in this scenario, the directions go both ways. Okay, so you can draw arrows in and arrows out of that cell. Now, I want us to focus on the water, remember? So if there was 90% salt, or sorry, if there's 10% salt in this scenario, that means that there's 90% water, right? Because, you know, 100 minus 10 is 90. So let's look at these next two examples where the movement won't be both directions, but it'll definitely be in one direction. So once again, a cell with 10% salt. Okay, so let's always remember in osmosis you want to keep the water in mind. So 90% water, okay, H2O. We'll shorten it up a little bit. So inside the cell is 90% water, and outside the cell is only 2% salt. So how much water does that make it then? Well, if it's 2% salt, that makes it 98% water. Okay, so I want you guys to look here and see which is bigger. Is 98 or 90 bigger? Well, I hope you all know that 98 is bigger than 90. So water always moves from where it has a higher concentration to where it has a lower concentration. So the water is going to move in to the cell because there's a greater amount of water concentration outside the cell. So that water will move in and that cell will expand. Okay, so can you think of how to do this last one then? So once again, inside the cell is 90% water, but now let's look at the outside. So if the outside is 30% salt, what does that make its water concentration? Well, hopefully you're able to do 100 minus 30 to equal 70. So now the water concentration is greater inside the cell. Water always moves from where it's greater to where it's lower. So tell me how it's going to move here, guys. I hope you guys got it. The water is going to move out of the cell. Okay? So I hope that makes sense. You'll see questions like this on the Sage. All right, let's take a look at some more current material. Number 21, order the levels of organization in the body from simple to complex. Pick one system. Don't use my same example, okay, in the body and go through that organization starting with simple, moving to complex. Okay, so let's do another example. I gave the example of neuron, brain tissue, brain, nervous system, human. Do you see how each level of that is getting more and more complex? So what do we start with? And you guys will write this on the lines, but I'll use my whole page since my marker's awesome. But the most simple type of thing we have is a cell, right? Our body is composed of trillions of cells. There's different types of cells. There's brain cells, those are called neurons. There's all different kinds of cells. So there's cells. What happens when those cells start to combine together? What do we call that? That is called tissue. Okay, so you have like skin tissue, right? And I want you guys to think of one other example that you could do. So we could do maybe a skin cell, right? So a skin cell would be an example, and of tissue, it would just be like a chunk of skin. I know that sounds gross, but that would be a piece of the tissue, chunk of skin. And then what comes after tissue? What do we have? We have an organ. And something unique about your skin is that that is actually considered your largest organ because all of your skin is one connected organ, right? And then you have your organ system, which would be the whole, like, it's called the integumentary system, which I know we didn't really learn too much about this, but integumentary Umentary system. And then you have the largest thing, which is called an organism. And that is just you. Okay, so that's the whole kit and caboodle. Okay. All right, guys. So I hope you enjoy my elementary school writing. But anyway, you could do this for any system in the body, but this is an example of how this breaks down. Cell, tissue, organ, organ system, organism. All right, this is something we just also recently talked about, is number 22, compare sexual and asexual reproduction by filling in the table below. So I bet you guys can help me do this. Number of parents for sexual reproduction, 2. For asexual, 1. Time to create new offspring. Now I'm not looking for something really specific here, but hope you remember that sexual reproduction takes a lot longer. Okay, and asexual reproduction is very short. Like bacteria that reproduce asexually can reproduce in, minutes. Whereas in sexual reproduction, like humans, for example, it takes 9 months to grow 1 new offspring. All right, how about the amount of genetic diversity that you get? So compare genetics from parent to offspring. Okay, so in sexual reproduction, we've often colored, if we could color, you get a mom and you get a dad that are different, right? And the outcome ends up being, so we kind of showed this with circles, the outcome ends up being a mix. So there is lots of genetic variation, okay? And in asexual reproduction, everything, parent equals offspring forever, okay? And the types of organisms that reproduce this way, for sexual reproducers, plants mostly reproduce sexually, and then many animals, right, reproduce sexually. There's 2 parents. And for asexual reproducers, definitely examples would be like bacteria, and anyway, that's a good example there, would be bacteria. And there's other ones too, but like a hydra, we talked about how a hydra reproduces by budding, and anyway, we talked about that in class. So anyway, that is the basics of the comparing sexual with asexual reproduction. All right, this one's an easy question, number 23. Give 3 examples of inherited traits and 3 examples of acquired traits. Inherited are things that you get in your DNA from your biological mom and dad. So it would be your hair color, color of eyes, it would be, the size of your toes, the funny finger shapes you have, or whatever, anything that's inherited, the way you look, or maybe a certain kind of condition you have, or something that also that your parents have. Anyway, those are inherited traits. And then acquired traits are the things that people can acquire later, like perhaps a good cook. That's a great trait to have. I wish I had a good cook living at my house. Maybe I could acquire that trait, huh? Or it could be a tattoo or earrings. That's an acquired trait. Okay, so you guys could probably think of 3 examples of each for you personally. All right, so we talked about this in class, about the fish. And so if you missed class, I'll do a quick run through of what it's all about. But I want you to use my example of the fish we used in class to describe the process of natural selection. So we know that species change over time. And the reason they change is because the environments will often change, okay? So we can sort of draw this example here and talk about under the sea. If we have, seaweed here, I'm just kind of drawing us a little scene here. Let's draw some Let's draw some yellowfish. Oh yeah, yellowfish. It's going to take a few minutes to draw this, but it'll help us. Let's draw some green fish. So we have different kinds of fish. Now they're actually in the same species, okay? But there are different kinds of fish, right? And then we maybe have some red fish as well. All right, so let's talk about this species. Right now, this species has about the same number of colors of fish. There's red, there's green, there's yellow fish, right? But let's say that there's some kind of storm that kind of mixes up the ocean a little bit more, maybe some kind of earthquake, tsunami or something. And now there is a predator that has not been in this water, but now is there. Ooh, yeah, let's draw Mr. Bleh. It's kind of a goofy looking, but this is a shark. So I want you guys to look at this picture and think about these fish. Do any of these fish have an advantage over the other fish? I want you to think about camouflage. I know you guys have read books about this stuff from being a young kid, but think about the fish, okay? So who might be targeted? Well, the red ones are probably going to be easy to be seen. Who gets to hide? The green ones hide very nicely. So even though all the fish might try to hide, the green ones are probably going to have the best chance of surviving. Now, does that mean that no green ones will, some green will die? Maybe that one dies. But overall, we see that in this population of fish, the green have a higher chance of survival. Now, when the green have a higher chance of survival, that means that they survive and reproduce. Okay, so it's not only important to survive, but it's also important to reproduce. So in the next generation, there will be more green fish because those are the fish that were naturally selected. So when we talk about natural selection, it's all about a species being better adapted to an environment to survive and reproduce. And through that process of natural selection, we have species kind of evolve and change. If we looked at this species of fish maybe like 10 years later, we might see that there are pretty much only green fish because the yellow and the red have just slowly been taken out of the population because they don't have a great chance for survival. Now, it could be that then again that the environment would change. Perhaps pollution in the water causes these seaweed to turn slightly more red or something. But then you can see how that it might shift the other way, and perhaps now the few red fish that survive, they have a better advantage in that environment. And slowly you could see that the population would shift towards, after many, many years, would have more red fish in it and maybe less green. So I hope that makes sense. And yes, I want you to sort of take what I said and put it into writing. So good luck. All right, you guys have got this. Number 25, Linnaeus developed a system for classifying living things. Give the order of classification starting with the most general and going down to the most specific. Well, we did come up with a little rhyme to help us remember. Do you remember? It was do, keep, wow, people coming over for good spaghetti. Do keep people coming over for good spaghetti. That helps you to know. Up here is the most general. Do you know what the D stands for? It stands for domain. That's the most general. There are 3 domains. That's the next question, I think. Then kingdom. And then phylum. P-H-Y-L-U-M. Class. Order. Anyone know what the F is? Family. Genus. and species. So that's right. It starts with most general, and as you go down the list, it gets more and more specific. And so every living thing has a classification. So like you and me, we have the domain Eukarya. We're in the animal kingdom. We're in phylum Chordata, and so on down the list. And our genus is Homo, and our species is Sapiens. So we are called Homo sapiens. That's our scientific name, developed by this scientist named Linnaeus, who kind of made a hierarchy of classifications. All right, guys, so I just want to make sure you guys know how to use the dichotomous key. So let's go over a few of these that you'll see on number 26. Always with the dichotomous key, you start with number, so like you're looking at a leaf here. Okay, so we're going to look at leaf A, and we're going to look at, we start with the top of the dichotomous key. So we read number 1 and we pick the option. So 1A is needle leaves, or B is non-needle leaves. Okay, so it's definitely non-needle. Okay, so that's A is non-needle. So we're going to go, so it's non-needle, so then we're going to go to 3, right? So then we go down here to 3. It's kind of like a find your own mystery. So 3 says it's a simple leaves, which means it's a single leaf, or is it a compound leaf, which made of leaflets? So tell me, look at A. Is it a single leaf or made of leaflets? Well, it's definitely a single leaf. So where are we going to go? We're going to go to 4. Okay, so we come down here to 4. Is it smooth edged or is it jagged edged? It's definitely smooth, right? So then we go to 5 and we read, is the leaf edge is smooth or the leaf edge is lobed? Is that what? I'm not sure quite what that means. So it says, yeah, sometimes we might have to look up words in here. So I believe it is smooth. It looks smooth to me, right? So I believe that this leaf is a magnolia leaf. All right, so we can go through this whole thing with all of these leaves, and that's what I want you to do. I want you to identify all of them. Let's do another one here. We can do B together. B, you know, is it needle leaves or non-needle leaves? Once again, non-needle leaves. Oh, so we're going to go to 3. Simple leaf, or is it a leaflet? It is a leaflet. So now we're going to go down to 7. Okay, so is the leaflets attached at one single point, or is it attached at multiple points? Well, I hope you can see that it's attached at multiple points, right? So B is going to be a walnut, right? So you keep these dichotomous keys are kind of fun because you guys can identify the name of like something without really even knowing what it is, but you can do it based on things you observe. So that's what's so great about this, is that because you can observe it, it's a lot easier. So anyway, and I did look up this here, the leaf edge is smooth or is lobed. I believe this is the one when they're talking about lobed. Is it still smooth? Well, anyway, it's smooth edged, but it's got like lobes in it. So when you get down to that one, this one's a white oak. So you'll see that when you get there. But anyway, I think you guys can do this. It's kind of fun. So make sure you know how to use a dichotomous key. Very helpful for identifying things that you may not know what they are, but you can observe traits of that thing. All right, I knew this question was coming. So all living things are classified into 3 domains. Write down each domain and a few characteristics of each domain. So let's break it down here. We've got the 3 domains. If you guys remember, it's Eukarya. Okay. And well, let's write them all. Let's write down the 3. We have bacteria. And this will be fun with all my nice writing. But you're almost done, guys. And then the last one is called archaea. A-R-C-H-A-E-A. Kind of a weird end. A-R-C-H-A-E-A. So let's do a couple things. First off, Can you guys tell me which of these are you? You are in eukarya. So most of the things in eukarya are multicellular. That means that the organisms in there are made up of many cells. Okay, so that would mean that archaea and bacteria, what would we call those? We call those single-celled organisms. Yeah. Okay, so these are single-celled. Now, the one other really big difference to talk about between these is we have some of these things that are eukaryotes and some are prokaryotes. Well, eukarya are eukaryotes. And what does that mean? To be a eukaryote, it means that there is a true nucleus in that cell. So that means that our cells and plant cells and any other eukaryotes, these multicellular organisms, have a true nucleus. What that nucleus is a special compartment for the DNA. Okay, and down here, these are called prokaryotes. P-R-O-K-A-R-Y-O-T-E-S. Prokaryotes, which means that the DNA is just free, floating. And that's not quite as beneficial for the organism because you know, it means that there's no protection. Like those directions for creating a new offspring are not like captured or not, you know, enclosed in a safety box like a nucleus is. Okay, so eukarya definitely they're often larger organisms. You know, these are macroscopic, and these down here are microscopic. Now you might be wondering, okay, well, what's the difference between these two then? If we're kind of lumping them together a little bit, well, the big difference is about where they live. So bacteria live everywhere. Okay, so I want you guys to put this in better quality writing than I'm doing. You know, basically you don't have to have it look like a kid, but an archaea, do you remember? They live in the extreme places in the world. So like you know, like volcano vents or deep sea ocean. The Great Salt Lake has some archaea, and they live in the extreme environments. They have something in their kind of like their cell wall that allows them to live in those extreme places. So this was a little busy, but hopefully you can pause it and write some of these things down. All right, we've made it. Last question. The question is how has technology changed our understanding of science? So things like cells, genetic engineering, classification, etc. So I think you guys know this, but you know, back a long time ago, people had no idea that humans were made of cells. Like we had no idea that we were composed of all these tiny, tiny little cells. So technology has allowed us to understand a lot of more things. So Robert Hooke, who was the discoverer kind of the cell, he was working with a microscope and realized that there were these tiny compartments that made up who, you know, like all these cells. So basically our understanding of science has grown and become more clear as technology has improved. So if we have better technology, then we are able to understand things. So like we used to just classify all things as either a plant or an animal. And once we got microscopes, we could see that there were all these like bacteria and microorganisms. So greatly improved our understanding, right? Genetic engineering, it's where we can like alter the genes in like a tomato plant or a strawberry plant to get a desired trait. All of these kinds of technologies, I mean, we've just really come to understand so many things. Our understanding of the atom, right? So we continue, and guess what? Do you think our understanding of science is still going to change? Yes, right? Because we don't know everything yet. And so as we continue to explore more and discover more and get better technology, our understanding of science will only get better. So that's kind of exciting. Thank you. All right, guys, you have survived. Thank you for watching. I know it was a little long. Hopefully you took a couple breaks, but don't forget to turn this in. Remember, you can either email me your answers, you can write your answers down and take pictures of like what you wrote, or you can just open up a Word document and write your answers in there as well. Thank you so much. Have a wonderful, wonderful day.